Hello everyone. So this class we're going to continue our topics of the fundamentals of environmental engineering and science. And this class we're going to talk about the risk assessment. So before we introduce the contents, let's again do a quick recap of our last class. So last class we basically introduced the ecosystems, right? So I want you to review these questions. For example, what is ecosystem? How do human beings affect ecosystems? Also, what are phototroph, chemotroph, autotroph, and heterotroph? And also, which one does produce the organic matter from the inorganic matter? They should be the photoautotrophs, right? And also, what are the aerobic, anaerobic, and facultative bacteria? What are the differences between bioconcentration, bioaccumulation, and biomagnification? Also, what are the solubility pump and the biological pump? What are the nitrification and denitrification? So what nutrient cycle are they being used? And also, where does the phosphorus in the surface water come from? And also, finally, where does most sulfur come from? Okay, so this class we're going to talk about the risk perception, assessment, and management. Okay, so the objectives of this work is to first, we should be able to assess the risk and then also to manage the risk. All right, so what is risk? We hear about risk quite a lot. So by definition, the risk is the measure of the probability to have an adverse effect. We're not really sure that it's going to cause the adverse effect, right? So that's why we need to calculate or need to define a probability here. And we're not saying that it's for sure that adverse effect is going to happen. So risk assessment means the estimation of the process of a risk. Right? So risk assessment is the estimation process of a risk. And risk management is the use of assessment results to make policy decisions. So in terms of the risk perception, it could be very wild, and it's more impacted by the personal experience and recent events. And actually, sometimes these risks might not be very long or might not be very strong. It's just that you or a specific person experience these risks, right? So how do we calculate this risk? So we have to consider what is the average or the lifetime for each human being. Let's assume that each human being has an average lifetime of 75 years. And then as further, we can find that based on the statistics, around 3.9 million deaths in the United States in 2001. Okay, and among these deaths, around 540,000 deaths were coming from the cancer. So how do we calculate the risk from the cancer? So the risk of dying from the cancer in a lifetime then can be calculated by the deaths that's coming from the cancer and divide by the total deaths in the United States. And you can find out that this risk is 0.14, which is around 40%. And this means this is the risk or the probability of people dying from cancer in a lifetime. Okay, so also people tend to convert this number into the annual risk. So therefore, based on the average lifetime of 75 years in total, we can calculate the annual risk by just dividing this 0.14 by 75. And we can get that the annual risk of dying from cancer is 0.002. Okay? And we should also notice that different risks will have their different effect. It can be accidents, it can be cancer, it can be smoking, so on and so forth. And for example, here is a risk or example about the risk of smoking. So smoking a cigarette is going to take 10 minutes, right? And then studies also found that it's able to shorten your life by 5 minutes. Okay, so this is the risk assessment. So what that means is that you basically waste 15 minutes to smoke a cigarette. Okay, so there are more recent studies about the adverse effect or the risk of smoking the e-cigarette. And as you can see, the death tolls are also increasing because of the e-cigarette smoking. And many teens, like young adults, are dying from the e-cigarettes. And because of that, the risk of smoking the e-cigarette is also going to increase. So we talk about the risk perception. So this is based on the larger data that we can calculate the probability of this risk here, right? And there's also the risk assessment, which is a more rigorous way to estimate the risk. So basically, that's a process that we can use to estimate the risk. And it involves the following steps. It includes the data collection and evaluation, toxicity assessment, and also exposure assessment. So the toxicity assessment means how toxic the chemical is, while the exposure assessment means how much contact we have. Okay? So in terms of the data collection and evaluation, it basically means that we're going to identify the substances of major interest through sampling and development of the sampling strategy. So in terms of the data collection, it can be first identify the possible contaminants, and also to understand what are the sources and the concentration of the contaminants in the environment. And further, we should notice that the environmental setting that could affect the fate, transport, and the persistence of these contaminants. For example, if the pollutant or these contaminants exist in the air or they exist in the groundwater flow, then they are going to have different data, right? So a quick example here. So the risk assessment will basically include all of these three, right? As we have introduced, they're going to include the data collection, evaluation, toxicity assessment, and exposure assessment. Okay? So then basically, how do we determine the relationship between a chemical and the adverse effect? So let's say we identified a contaminant and we want to calculate or want to assess the risk of this contaminant. Okay, so we're going to do the following things. We can first conduct a hazard identification. Basically, that means to determine whether the exposure to a contaminant causes the increased adverse effects for humans and to what level of severity. And we can also do animal tests. Okay, so we can conduct these tests on species that are other than human. Okay, so we can also study the dose-response evaluation. So basically, this is the derivation of the relationship between the dose. So basically, dose means milligram per kilogram of the body weight, and the incidence of the adverse effect or the adverse reaction. And typically, we should notice that the dose-response curve is going to follow a Gaussian distribution. And we can also define a parameter, which is called LD50, means that means the lethal dose for 50% of the test animals. So what that means is, let's say we are trying to apply certain contaminants. We're trying to study the toxicity or the risk of mercury in water, right? So what we can do is we can set up a range of concentration. Let's say 0 milligram per liter, and then 10 milligram per liter, and then 20 until, let's say, 1,000 milligram per liter. Okay, so then we're going to we're going to conduct experiments on animals to determine the risk of the mercury by feeding these solutions or feeding these water to different groups of animals, right? Let's say we want to use rats as the test animal. Then let's say we have 10 rats for each group, right? Until 1,000, 10 rats. And then we're going to basically generate a curve here. So what we can see is that as the mercury concentration increases, then the lethal rate, okay? So lethal rate is also going to increase. It's going to follow a curve like this. Okay? Lethal rate or death rate. So basically the LD50 means 50% of the test animals are dead because of this contaminant. Okay? So we can basically find out what is the LD50 based on these experiments. Okay, so here is actually showing another way of these types of experiments. Let's say we have compound A and compound B, and we're testing them on animals to try to understand the risks of these compounds. You can see that the compound A actually have a lower LD50 than compound B. So then I have a question. Between A and B, which one is more risky? It should be A, right? That's mainly because for A, you just need a lower concentration to reach to 50% of the lethal dose. So basically, the lethal dose corresponding to 50% of the mortality is lower for A species. This means that the compound A is actually more risky. And you can also see this symbol or this acronym here. So basically, this shows this value represents the lowest concentration or the highest concentration of the contaminants that are not going to pose an influence on the test animals, right? So if you see, let's say for the compound A, then this value here, which is around let's say, which is around 1.3 here, okay? So this means that if this compound A concentration is below 1.3, then that's not going to pose any risk, or the probability of this risk is negligible, okay? And similarly for compound B, this value is around 6.5, okay? So this value, NOAEL here, represents the highest concentration of these contaminants that are not going to pose a significant influence or perceptible influence onto the test animals. Okay, now let's say, what does LD50 mean? So we have two definitions. One definition is the dose of a chemical that causes 50% of the testing species developing 6 symptoms. And the B represents or tells us the dose of a chemical that kills 50% of the testing animals. So the answer should be B, right? Because this is a lethal dose, and lethal dose, the lethal means that there's going to be mortality there. All right. So as we have introduced, the NOAEL is actually short for the no observed adverse effect level, and this is specifically representing for the animal test. Okay, so NOAEL represents no observed adverse effect level. Okay, and for human beings, we're going to use this NOAEL divided by an uncertainty factor to represent this similar quantity called the RFD. Okay, this is mainly because humans are going to respond differently compared to the animals. And also, we have more definitions. For example, the acute toxicity. So acute toxicity means rapidly develops the pronounced symptom. Okay, so the symptoms are going to show very rapidly. And there is also chronic toxicity, which means that the toxicity or the risk is going to take a long time to develop the symptoms. So one is short-term symptoms, one is long-term symptoms. And as we have introduced, we actually use these animal tests to find out the risk to human beings. And therefore, there are limitations of these animal tests. So first, animals are not human. So because of that, the responses of these contaminants are going to, or responses of human to these contaminants are going to be different compared to the animals. And also, the animal tests are normally occurred in a short time, okay? But for humans, the latency period is normally for many years. And this is, again, for these experiments, let's say we want to understand the toxicity of certain species, we'll just conduct, let's say, 1 year, 2 year, or 3 year at most on test animals. And also, these animals normally have a shorter lifetime, right? But if you consider influence on human beings, so human beings have a average lifetime of 75 years. Who is going to conduct experiments over this long period of time, right? So there's no way we can do these long-term measurements or long-term evaluations. So that's why the animal tests, because they're short, they're going to pose limitations. Okay, and furthermore, the animal tests are normally for a single chemical. Let's say we want to understand the influence of mercury on the animals, right? But for humans, there are many complex factors, including multiple chemicals are being involved. For example, there are incidents that the mercury concentration becomes higher in the river, and that's polluting the drinkable water. But not only mercury concentration is high, but there are also other heavy metals or other pollutants existing in the drinking water, right? So there's going to be a complex system that's affecting the toxicity of these contaminants on human beings. And we as human beings are not only, there's no way we can just drink water that has different levels of the different concentrations of the mercury, but not include but not including the change of other species. Okay. And for the risk assessment, people also conduct the epidemiological studies. So for these studies, it typically involves the large population. Also, it includes a long latency period. We also try to consider the mixed factors. But one thing we should notice that is that the epidemiological studies, typically for these data collection, they may not be representative because for these very long period of assessments, there's no way we can include so many samples because there's limited time and efforts from these experimentalists, right? So another quiz question. So why can't we directly apply animal test data to human? Right? Is it because the animals can tolerate greater chemical impacts? Or is it because data from the animal test are short-term and often from a single chemical? It is because of B, right? And for example, animals can be different. Let's consider rats as an example. So rats are very small, right? So because of that, the lethal dose is going to be lower compared to human, simply because human have a larger body mass, right? So because of that, we're more tolerate to some chemicals compared to the rats, all right? So the third part we want to focus on is the exposure assessment. So for the exposure assessments, we want to understand how these chemicals can interact with a human being, okay, or interact with the test animals, okay? Because for different contaminants, they may enter our body system by different ways. For example, if we want to understand the total exposure assessment, for example, let's say we want to understand how human beings are exposed to mercury, then we want to consider the exposure by considering all of the following pathways. For example, we want to include the groundwater, so mercury can exist in groundwater. It can be in surface water, right? They can exist in the sediment, they can exist in the air, soil, dust, and food. So we want to basically calculate a reasonable maximum exposure, which is basically the highest exposure that is reasonably expected to occur. Let's say we keep drinking water, right? When we swim, they can get permeated into our body system, right? When we breathe in, then these mercuries can get attached onto a particular matter. They can get in our respiratory system. And if they exist in the food, we can also eat them into our body system too, right? So because of that, for different pathways, there's going to be a different calculation method, okay? So basically, we can calculate a chronic daily intake, which basically means the milligram of this contaminant per kilogram of the body weight per day. So we're going to calculate this value here, and then basically calculate this value from all of these pathways, okay? So this is the equation that's being used for, this is basically an universal equation to calculate these for these different intakes. You may see that it's actually quite complex here, but as a matter of fact, if you know what are the basic understanding here, right? Let's say, finally we're going to calculate the milligram of this contaminant per kilogram of the body weight per day, right? So these are going to make sense. So the chronic daily intake, or the CDI, can be calculated by C, which is a chemical concentration over the contact period. You can just think of this as the chemical concentration of this contaminant. Okay, so the CR is the contact rate, or the EFD is the exposure frequency and duration. Okay, the BW is the body weight, and the AT is the average time. Let's say, for example, we're drinking water. Okay, so we want to understand what is the risk or what is the chronic daily intake by drinking water of the contaminants, for example, the let's say mercury again. Okay, so first, if we want to calculate this, we need to know what is the concentration of the mercury in the water, right? So the C here is going to be the concentration of the mercury, all right? The CR, or the contact rate, so basically means how much water you drink for each time, okay? Let's say you drink 1 liter water per, let's say, each time, okay? 1 liter water, or 1 cup of water each time. And the EFD here means how many times you drink this water over, let's say, 2 days, all right? Let's say you drink 10 cups of water, or let's say 1 cup of water each time, and you drank 10 cups of water for 2 days. All right, so the EFD here is going to be equal to 10, and here is equal to 1 cup, but we need to consider the volume, right? And BW is body weight, and the AT is the average time, which is 2 days. So now if you consider what that really means here, okay? So we drank 10 cups of water that's containing mercury, right? So what we're going to do to obtain this chronic daily intake, or CDI here, is just consider, or to just calculate the total mass of the mercury, divide by the mass of the body, over 2 days, right? And the mass of the mercury is just 10 cups of water multiplied by the concentration of the mercury in there. Okay, so this is quite straightforward. So as a matter of fact, if we want to calculate the ingestion from the drinking water, people develop these more complex equation here, but it's still, if you consider the entire process, it's going to be the same. So the CW here means the chemical concentration in water, right? It's just like the mercury concentration in the water we discussed. So the ingestion rate, or this IR here, represents how frequent or how much the water we're drinking for each day. And the EF is exposure frequency. Okay, so basically, if we want to consider the polluted water here, okay, so we're not drinking we're not drinking water with a lot of mercury each day. It can be every month or after a week, we're going to get exposed to these high levels of mercury, right? And then the ED here means the exposure duration. So basically, for how many years we are exposed to these high concentration of the mercury. Of course, we need to divide by the body weight, and then divide by the average time, okay? So it means, let's say we want to track over 2 years, okay, the high concentration of the mercury. Then the average time is going to be basically how many days over these 2 years, okay? So as we have introduced, these contaminants can also get permeated into our body through the dermal contact. Let's say when you are taking a shower or when you are swimming, all right? So they can also penetrate through our body system, all right? So this is calculated in a different way. You can see that CW is again the concentration of this contaminant in water, all right? The SA here is the skin surface area, because we're taking a shower or swimming, right? The PC is the permeability constant, all right? Because not all of these contaminants are going to quickly permeate through our skin there. So each of the contaminants, they're going to have a specific permeability constant, okay? So the ET is exposure time again, right? So how long do we take a shower? How long does it take for us to swim there, right? And the EF again is the frequency, right? And then this the ED is the exposure duration, right? CF is the volumetric conversion factor for water, okay? Because we're basically converting this skin contact, which is in the unit of centimeter square, into liters here, because we want to make sure that the unit will cancel, or unit can be unified, right? Again, it's dividing by the body weight and then dividing by the average time. So you can see, actually, when we have an example, it's going to be more straightforward. So we're not going to always follow this type of calculation there. And sometimes the calculation can be quite straightforward. Okay? So there are also other exposure pathways. For example, it can be ingestion while swimming, right? Drank a lot of water during the swimming. Ingestion of chemicals in soil. It can be dermal contact with the soil, right? When we're playing at the beach, right? Inhalation of airborne particles. This is related with the air quality. And also ingestion of contaminated fruits, vegetables, fish, and shellfish. Okay, so the shellfish are also going to accumulate these heavy metals or contaminants. And we have to separately consider the risk from these contaminants. All right, so let's do a quick example here. So it wants us to calculate the chronic daily intake, or CDI, of the benzene, right? So as we know, so the example basically tells us that benzene has a concentration in water of 0.005 milligram per liter, okay? So we drink 2.3 liter per day for 63 years. Okay, so we basically want to calculate what is the daily intake of the benzene, right? So, for example, so based on the condition of the first two, we can calculate what is the CDI coming from drinking water, right? Because we drink for 2.3 liters, right? And then we drink at a concentration of what? 0.005 milligram per liter. Okay, so this is going to give us a mass of this benzene over each day, right? And then what we just need to do is divide this number by the body weight. Okay, so body weight is 75 kilogram, right? So then we can calculate the CDI that's coming from drinking the water, okay? And so there's also thermal contact by this water here. So we swim 3 times per week and 0.5 hours, basically 30 minutes per time for 45 years, okay? So what we need to do is, again, to basically calculate what is the surface area of the human body, right? And then multiply that by the concentration of the benzene, right? And divide the the body weight of the human again. And also we need to consider what is the thermal contact or the thermal uptake from the water, okay? And there are also conditions that during the shower we can also ingest these contaminants, and there's a condition that only 1% of the available benzene is absorbed during the shower. And here we have the skin area, and then we can calculate what is the CDI from these different ways. Okay, and of course we need to consider the breathing of the benzene from these breathing process. Okay, so basically there are 4 ingestion methods. Okay, and then respiration from breathing, right? And to calculate the CDI, we need to consider all of these ingestion processes. Okay, so for the first one, you can see, so basically you have ingestion by drinking, right? You have ingestion by contacting with the shower, right? You have ingestion by swimming. So based on that, we can calculate the CDI from different ingestion method. So first one, from the drinking ingestion, what we can calculate is, so first we have a concentration of the benzene in water, right? We drink 2.3 liter per day, and then there are 65 days per year, and we're drinking for 63 years, right? So in order to calculate the lifetime or the chronic daily intake, we need to consider the lifespan of the human being too, because the lifespan is 75 years. Okay, and then because of that, we can calculate what is the daily or the CDI from the drinking ingestion, and that turns out to be 1.24 multiplied by 10 to the minus 4 milligram per kilogram of body weight per day. Okay. So we can also calculate the showering absorption. So in terms of the showering absorption, we first, again, need to use this concentration of the benzene in water, right? And we're going to multiply by the, what is the volume, basically what is the surface area of the human being, and then multiply that by this absorption rate, right? It's a penetration rate. So for each hour, this benzene is going to penetrate 0.002 meter, which means that it's 2 millimeters through our skin. And there are, for each event, we have 0.5 hours, right? Each event, each day there's one event, and there's 365 days, and then there are 63 years, right? This is based on the average time for 63 years. And again, in the denominator, we need to divide by the body weight and the average time, which is 75 years. And that turned out to be 1.04 multiplied by 10 to the minus 4, okay? And the condition also give us that so only 1% of the available benzene on the skin are absorbed during the shower, okay? So this means that we can just multiply this number by 1%. And actually the actual the CDI is just 1% of the ingestion rate. Okay? And then based on similar method, we can calculate what is the CDI coming from the swimming absorption, the inhalation, and during the inhalation during the showering process, and also the ingestion while we're swimming. Okay? So finally, to calculate the CDI, we're going to basically take the sum of all of these terms, and this is going to be the final CDI. And you can see that the drinking process is a major process that's leading to the risk of these contaminants, right? What is the second most significant one? It's actually the swimming absorption. Okay? And of course, the inhalation during the showering is also going to affect. All right? So furthermore, there are definitions of the low cancer risk or high cancer risk. So basically, the low cancer risk is calculated by intake multiplied by the slope factor. So slope factor is a unit risk per unit dose. Let's say for benzene, after we calculate the CDI, we can multiply this risk of this benzene to calculate the total risk. Okay? And for high cancer risk, we can calculate based on this equation. So 1 minus the exponential of the minus intake multiplied by the slope factor. So basically, the higher the intake, The larger the intake, you can see that larger the intake, the smaller this exponential term, right? And y minus that is going to give us a higher risk, okay? So the total risk is calculated by taking the sum of all of these different risks. And for benzene, the slope factor is 0.029 kilogram per day per milligram, right? So if you already have these calculations here, and then you can multiply the CDI by this slope, and that's going to give us 4.9 multiplied by 10 to the minus 7. So what does this number mean? It means that for every 10 million people, which is 10 to the 7th number of people, just 5 people die of the cancer from benzene in a lifetime. So this is still a very small number here, all right? So there's also non-cancer risk. Non-cancer risk, for example, the hazard index for HI is calculated by the intake divided by the RFD, and the total hazard index is calculated by summing all of these hazard index together. Okay, so another quiz here. Which pathway is considered in the total exposure assessment? The exposure through drinking or contacting water, exposure through uptake of dust, exposure through breathing, exposure through dieting. Actually, it can be all of them. It can be ingestion pathways from all of these processes. Okay, so finally is the risk management. So risk management means to determine the magnitude of the risk that can be tolerated. Okay, so we can manage the risks by changing the environment, to modify the exposure, basically to reduce the CDI, right? If you can reduce the CDI, then that's going to reduce the risk. And also to compensate for the effects. For example, it's like taking the antidote, right? While you drink the toxics, you can drink the antidotes to compensate these toxic or risk processes. So let's do a summary. So basically, during this lecture, we learned about the risk perception, assessment, and management, right? So risk perception is based on the existing data and experiences, okay? So based on the death toll coming from the car accidents or coming from the cancer, we can calculate what is the risk. And a more rigorous way for assessing the risk is done by data collection and evaluation. So we need to consider the representative sample, right? So for toxicity assessment, we need to, for example, want to know how toxic the chemical is. We need to calculate, basically to access all of these. We will conduct the animal test to determine the LD50, right? And for the exposure assessment, we're going to consider all of the pathways. And can include drinking water, can include the breathing, basically inhalation, showering, right, swimming, so on and so forth. And in terms of risk management, which is to reduce the risk, we can change the environment, modify the exposure, basically limit the access, reduce the CDI, and compensate for the effects. Okay, so let's review these contents by looking at these questions. So what are the 3 aspects of the risk assessment? It's already shown here in the title, right? And what is a toxicity assessment? What are the limitations of the animal test? We introduced on that too. What is NOAEL? So basically this is the maximum concentration that did not lead to observable risk to the test animals, right? Also, what is LD50? What is exposure assessment? And how do we calculate the CDI for a certain chemical? Also, how do we calculate the risk of carcinogen at low and high cancer risks? We're going to use different equations to, one is to directly take the product of the CDI with the slope. The other is to take the residue of the exponential term there, right? And also what is the hazard index. So that's all for the contents of this class. Let